hide well?
Yeah.
Okay, perfect. Thank you very much to the organizers for inviting me and giving me the possibility to share some recent work that came out of my lab, where basically what I try to show you are some results that illustrate how if one is very careful in keeping in mind the physical environment really in which a population expands, can find out that viral populations, specifically in our case phage, but this I think also applies to viruses expanding, like for instance in a tissue, are subject to an inevitable density dependent dispersal, and how this actually generates pushed waves. Okay, well, I think we would all agree here that spatial range expansions are ubiquitous, both across different species as well as scales. So I think we are all very familiar with the megaplate, where it was shown how quickly E. coli populations can gain resistance to antibiotic when these antibiotics are distributed in space. Another famous example are the cane toads, and similarly for flies in Australia, that over the course of their range expansion through the continent evolved in order to migrate faster and faster. Obviously, these spatial range expansions also apply to viruses, and we're currently experiencing the latest of this. This is an example from a paper from last year that showed the first 30 days or so, the first wave of SARS-CoV-2 spread in Italy. So we can see how quickly it spreads over the whole country. Right, so in the past, while I was a postdoc in Oskar Halacek's lab, I worked quite a lot on microbial range expansions, especially to quantify the genetic diversity in colonies, which are crowded cellular populations, but also the effect of selections. Towards the end of my postdoc, I kind of shifted a bit my interest towards viral range expansions, with a specific focus for our model system into phage, T7 phage. So T7 phage T7 is a double-stranded DNA phage with a capsid of around 50-60 nanometers and a short tail. Its life cycle is very simple, since it's a forcefully lytic phage. It just adsorbs to the bacterial host, injects its genome, the genome starts replicating, and also the proteins that will go on to form the capsid and the tail are transcribed, translated, and assembled inside the host until host lysis and release of the new phage particles in the environment. So also these viruses can expand in space, and the equivalent of what a microbial colony would be in phage is a plaque or clearing, which is a picture here. So this is an example of this clearing, where we have around a droplet that represents the lawn of the host, and we see from the initial inoculation the phage expanding in space and killing the bacteria as they grow. So what we really observe here is the death and the absence of bacteria. So something that really triggered my interest was an evolutionary experiment that actually Wolfram Möbius, who is now at Exeter, did again in Oskar Halacek lab, where it was trying to evolve phages over a range expansion. So the basic idea was that it would inoculate some phage on an initial bacterial lawn, let it expand for around 17 hours, pick at the edge, and re-inoculate again on a fresh lawn. And he kept doing this over several days, 14 days in total. Now, the expectation, both from theory and from previous microbial experiments, was that we would see expansion load, so the accumulation of deleterious mutations at the edge of the expansion, which is what is expected, given that in these range expansions, the genetic drift is very strong because the effective population size at the end is very small. So one would expect selection to be very inefficient. And indeed, this was shown in microbial experiments that were conducted very similarly, where a droplet of bacteria were inoculated, let it go for a while, picked at the edge, re-inoculated, and so on, and over like 30 and more days, in general, the colonies will grow smaller and smaller, showing this decrease in fitness. However, what actually Wolfram observed in his experiments was an accumulation of mutations that were actually quite consistent across replicate. But in particular, what I want to highlight here is that the front velocity kept increasing in the course of the experiment. These phages were getting faster and faster, which is very different from what instead was seen in the microbial example. So what is different? Well, clearly there are many reasons why this can happen. So there may be a different availability of mutations in the phage case and in the microbial case. But something that I kept batting my head against was whether there was something very intrinsic in the viral case compared to the microbial case that would cause this difference. And one particular property of the traveling wave that can limit the expansion load, so the accumulation of these deleterious mutations, is when the traveling wave is pushed rather than pulled. So just to recap, the difference between pulled and push waves is that in the pulled wave, most of the growth occurs at the very leading edge of the wave, which is pulling the wave forward. Instead, in a pushed wave, the maximum growth occurs a bit behind the tip of the wave when the density is at intermediate values. And this normally happens because of cooperative effects, for instance, an alley effect. This difference in pushed and pulled waves has quite dramatic consequences when it comes to genetic diversity. Because if we imagine that we start from a population that is all diverse, neutral, in the pulled wave, the probability that an individual will fix later in time becomes larger and larger the closer this individual is to the front of the population. In a push wave, that's not the case. Actually, the individuals that have the highest chance of fixing later are somewhere behind the wave at a specific, in a specific region that depends on the strength of pushness of this cooperativity, in fact. And this can be was actually very well quantified by Gabriel Birzo and Kirill Korolev a while ago, where through simulations and analytical arguments, were able to actually quantify an effective population size as a function of cooperativity, where it shows very well here that as the cooperativity increases, meaning that the wave becomes more and more pushed, the effective population size gets larger and larger. Now, if we go back to the experiment, the phage experiment that I showed before, it's clear that a very large population size will make also fitness selection more efficient. So this is why it would limit actually the occurrence of expansion load. So what does this mean? Are viruses actually cooperating, and why would they do that? So that's really the question that I kept coming back to. Well, let's look at how actually, for instance, viruses, like in the case of phage, expand on a bacteria alone. So this is quite, old models that have been looked for a while. So the idea is that we have three different species. We have the free diffusing virus, we have the uninfected bacteria, and we have the uninfected bacteria. When virus meets bacteria with a certain rate, the bacteria becomes infected and the virus absorbs the bacteria. And then after a certain period called lysis time, the infected cell lyses and beta new viruses are released. So if we are now in a spatial environment, oh, sorry, I forgot to say, the beta is called birth size. So if we are now in a spatial environment, we can write a system of partial differential equations that describes the dynamics of the uninfected bacteria, the infected bacteria, and of the virus. And here we're assuming as it is often the case in this bacterial lawn, that the bacteria are non-motile, so they're not moving around, which is why there's no diffusion associated with the bacteria, and only the viruses can disperse. So something that is important here to point out is that the diffusion coefficient here of the virus can actually be dependent on space. Like it may not be constant in space. And in particular, what I'm going to show you in a second is that this diffusion coefficient actually depends on the bacterial density, so on the host density that the virus has around. All right, so what we started working on specifically my student, Michael Hunter, really worked on was trying to quantify how the viral dispersal depends on the host density. So if we have this lawn of bacteria, how much the dispersal of the virus will be affected by this. And we can think that this lawn of bacteria is actually a very crowded environment. There's a lot of hosts out there. So there are even steric interactions that can hamper the dispersal of the virus. So to test this, he designed the following experiment where started with a plate. He covered the plate with a lawn of bacteria that were, however, resistant, meaning that the virus cannot infect this background host. And on top of this, he inoculated several droplets of phage and susceptible bacteria. So the point of doing this is that the background bacteria, because it's resistant, just functions as obstacles really to the diffusing virus and doesn't affect the infection dynamics of the virus at all. And we can use the susceptible host droplet to actually measure the time it takes for the first virus to diffuse through this background bacteria and get to the susceptible host where we will see a clearing appearing. So if we plot the distance between each droplet as a function of the time in which we see then the clearing appearing, we can confirm that indeed the behavior is diffusive, but we can also infer a diffusion constant for all the different possible background bacterial densities. And he repeated this with different bacterial density and he found a very nice relationship of diffusion of the virus as a function of this background bacteria, which was something that was hypothesized in the past, but it was nice to actually be able to quantitatively and experimentally measure it and actually put real parameters in this. So what we find is, according to these experiments, is that around 2 bacteria per micrometer square, the diffusion of the virus should almost completely halt. So if you have very, very packed bacteria, it will be very hard for the virus to actually diffuse around. So now that we have this diffusion, we can actually put it back in our system of 3 differential equations, solve it, even just deterministically, and see what happens. So what you predict, whether you have this actually density-dependent diffusion or not, is that over the course of the expansion, you will create 3 waves. One is the viral wave that will expand with a certain velocity c. And just associated to it, there is a bacterial wave that instead recedes at the same velocity. And the region in between of infected bacteria with a characteristic length scale here called delta x. So we see this kind of behavior for different variants of the model. In particular, we looked at 4 variants which were very meaningful, both for phages, but also for general viruses that expand into tissues. So the first one, which is the difference between the top row and the second row, is the effect of including this density-dependent diffusion. So the idea that because as the wave of the virus proceeds, and so the bacteria recede, there will be an inhomogeneous bacterial density that then will generate also an inhomogeneous diffusion, dispersal of the virus, so that the virus in the bulk of the wave is actually able to disperse faster than the virus that is at the tip of the wave. And we wanted to compare what basically the introduction of this point, how the introduction of this point affect actually the result of the model. The other thing that we looked at, which is in this case like left column versus right column, is what happens if we allow the virus or not to actually infect previously infected cells. And the reason why this is interesting in the case of phages is that we find in nature different kinds of phages. Sometimes they do allow infection of already infected bacteria, otherwise they don't. Other times they don't. So it's interesting to see what this effect also will show. All right, so something really nice that Gabriel and Kirill showed in their paper was that actually one can find out whether a wave is pulled or pushed simply by the deterministic velocity that you find in the wave, meaning that you don't have to account for stochasticity. You can just solve the system, for instance, of partial differential equation, measure the velocity, and compare it to the velocity predicted by a linearized approximation. And in this way, we can get exactly transitions from pulled to what they call semi-pushed to pushed, which in this plot over here are represented by different gray regions. So we started with just a typical numbers for lysis time and absorption rate of the for phage T7. And what we found is that the different model variants actually vary quite a lot. In particular, what I'm plotting here is the velocity of the front as a function of bacterial density. And what we find, at least for the case of T7, is that as long as the diffusion is constant, then the phage pretty much propagates with pretty much a pulled wave, we can say. But as soon as we introduce this variable diffusion, instead we get a high density that actually transitions to a push wave. So it can transition to a push wave. And this effect is stronger when the viruses cannot infect previously infected bacteria. So we can repeat this across different parameters. So what I'm plotting here for the 4 model variants are this transition from pulled, which is light gray, to semi-push, which is a bit darker gray, to fully push, which is very dark gray, as a function of bacterial density, and this product here of absorption rate by lysis time, which our model finds to be the key parameter to actually predict this transition. And what is interesting here is that with the exception of the first model, this UDM+, all the others show regions that are in push, that can be pushed. And just to give you a reference, like the experiments that we did, the experiments that Wolfram did, were more or less in this region. So in this case, it would be a VDM+ model that applies to T7. So they're clearly in the push.
Diana, it's frozen for me. I'm the only one. Am I the only one?
No, she froze. The connection was bad.
Okay, let's wait.
She'll probably come back in a second.